As far as plant structure, there was like the different levels that you could look at was like cells. And so when you look at cells, how many different cells were there that we discussed? Yeah, 3, and then there's actually more than that. But I always like the rule of 3 on this. There's parenchyma, right? And then there's what else? Perfect, parenchyma. And then there's parenchyma. And that's like the normal 3, but then you also have like the specialty 3, which is going to be, or the specialty other ones, which are going to be like your vessel elements. And the tracheids, which collectively form what? Okay, I'll take it. It's the xylem. And then there's also the sieve tubes and their neighboring companion cells, which form the what? Okay, I'll take this one as well. The phloem. Yeah, right? We remember that, correct? From back in the day? Now, what do cells create? Cells create organs, right? Yeah? Tissues. How many tissues? No, not those tissues. Sorry. How many tissues would you see in a plant? Different types. 3. What are they? Dermal. There's ground and vascular. Perfect. And what's the vascular tissue containing? What two types of systems? All right, xylem and phloem both. It worked out like magic if you think about it. Whatever. And then after tissues is what? Organs, correct? What are the 3 organs that you would find in a plant? Roots is one, good. What else? Leaves. What? Stems. Or shoots. How come people say chill out? Why don't they ever say chill in? What did you try to chill in? What types of tissues would you find in roots? What? Well, then what types of tissues would you find in leaves? Vascular? Dermal? So ground tissue is exclusive to roots is what we're going for? These tissues are, all 3 of them you can find in each one of the organs. You remember that, right? That was all good stuff? Perfect. Another thing that I needed to talk about from today was this idea of transpiration. Is transpiration the pull or the push that you see with water in the plant? So this is going to be more of the pull. What's the push that's created? Perfect. Root pressure. So root pressure would be more of the what? The push? Okay, so transpiration is the pull, correct? Now what is transpiration just naturally, like if you were to define it? Loss of water, right? Through the what? Okay. What's the stomata typically supposed to be losing or gaining? It's all about gases, right? But in this case, there's water that's escaping out of it. Correct? Now, what else do we know off to the side about water? Just like is this unique fact about water? Do what? High specific heat. What else? Evaporative cooling. Surface tension. Okay, adhesion, cohesion. That's where I want to go. What is adhesion? Okay, so sticks to others, and then cohesion is going to be sticks to self. So you have like these 2 things that are helping water stay basically connected to about everything that it's around, including itself. And then you're pulling it. Not only pulling it, but there's root pressure, which is this idea that you have most of water, or you would expect to have tons of water around the roots. And that's what's pushing water into the xylem. In other words, the plant is likely to take in water, because water is going to go from where it is to where it is not, which is going to be from the soil and around the roots, and it will go to inside the roots. It just works out this way. Cool? Awesome. Let's see. As far as like controlling your stomata, because I think that excuse me, I think that's something that we need to talk about. Let's talk about the stomata real quick. What's the name of the ion that's making this possible? So this is all about potassium. So the mechanism of potassium ion control. Now it would make sense that you have like the stomata, which again is this opening, and on opposing sides of the stomata are these two cells. Now I'm drawing them pretty poorly, but it's whatever. What's the name of this guard cells? You know what I mean. The names of those cells are the guard cells, which are going to open and close the stomata effectively, right? Now it would make sense that when you have stomata closed, that would be a case in which water has moved out of the guard cells, right? So if water moves out of the guard cells, what happens? The stomata is going to close. That means the stomata are going to come in here and like blockade this section. Now that would make sense. Do you see the two lighter circles that I've drawn right here? What happened to those guard cells? They got smaller, right? They got smaller and they covered up the opening. How would you get smaller? How would you take them from this large size to this small size? You know, water's got to go out. Now if water's going to go out, how would you force that event to happen? Of what? Of potassium on the inner out, on the outside. So high K plus concentration on the outside causes H2O to move out of the cell. So water moves out of the cell, right? So here's your guard cell. There's a lot of my little dots, which are the K plus particles, and water goes out. Now, after that happens, tell me what happens, or what the guard cell looks like after that event has happened. You know, your guard cell is all like shortened, small, and spots of water, correct? Would you say that the guard cell is in a hyper or hypotonic solution with respect to the potassium concentration? Hypotonic? So the guard cell is in a hypotonic solution? What does hyper mean? Too much, right? This solution that's around the guard cell is too much water release. You're in a hypertonic environment. Now, what if I ask you about the cell? Is the cell, in this case, is the cell hypo or hypertonic to the solution? You see the reversal, but like it's preposition different. So it's like, who are you talking about in relation to something else? The cell's hypotonic. The solution is hypertonic. Make sense? Okay. The cell shrivels up, and obviously the stomata is covered in this case. It's like saying that you put tape over your mouth, essentially. Can you breathe? No. Well, okay. And tape your nose too. So no breathing, right? So this is like, this would effectively stop the removal of stuff into and out of the stomata, right? And it's not just the gases. Now we're seeing it's also the water thing too, right? So you close the stomata, and yes, gases stop moving in and out, but at the same time you stop losing the water. That's the idea of C3, C4 in can plants, which is why you got that packet. Cool? Awesome. What is the stem cell? Okay. What is the idea of differentiation then? So a stem cell is like the ability to differentiate, whereas differentiation is the product or it's the event of becoming like specialized. Pluripotent? Mm-hmm. So stem cells are, maybe we could view this as being like a blanked out cell. A blank cell? What does that mean to say blank cell? What? Yeah, non-specific. It's not determined, right? It's not having a specific function. It is able to become whatever. What is differentiation? That's the process of becoming not just a whatever, but becoming a specific focus, right? So it's a process that focuses a cell with one, maybe the word goal would be good there, or one function. Do you have stem cells in your body right now? Bones. Because those cells could still become B cells or T cells, so technically, right? But you would have more stem cells like when you were first formed and everything was ready to be differentiated, correct? Now, what does that mean? Like, what limits does that impose for you? If your cells become determined and they become very specific over the course of your life, they lose this ability to go wherever they want to. That means that there's a point in time where your growth stops. You could agree that as differentiation increases and your cells become more determined, you stop growing more and more and more. Right? Now, you know that plants, though, don't have this determination everywhere throughout their body, correct? Or their plant body. So they have meristems, which is going to be a region of growth. And obviously it's made of stem cells. Now if you look at some different types of these would be like an apical shoot meristem, or maybe an apical root meristem, or maybe a lateral meristem. The term meristem is making reference to the fact that it's embryonic tissue that could differentiate. Apical versus lateral is kind of talking about the growth. If you think about it, your apicals are always going to be about primary growth. Your lateral, of course, is always about secondary growth. So I put primary by apicals and I put secondary by lateral. So if you're going like through the age of like 12 to 16 and you grow taller, would that be a primary or secondary thing? That would be primary. You're growing taller. Now, as you grow like more round, and that's what happens to plants basically, as they grow more, they're going to grow more round. That's going to be a lateral growth. That's going to be secondary. Cool? So you could say that this is more of growth in girth as opposed to growth in the root. This would be downward growth as opposed to upward growth. Right? We talked a lot in this case just about apical meristems, and that actually brings up a unique point of something called apical dominance. Do you remember what apical dominance was? What that resulted in? Exactly. If that meristem is the one that's receiving most of the chemicals. So it's a fact, or it is this concept that the apical meristem is more active, you could say, than other meristems. And the reason that, and Matt had just alluded to it, the reason you have this meristem on more, or more reactive, or doing more work is because it's going to do a direct result, or it's going to result directly in what? Like tallness, right? So you have growth upward, which of course is for one reason, and that's going to be a reason of light. Now, what if you remove those apical meristems? Those chemicals that cause cells to grow are not going to end up with that meristem because it's absent. They're not attracted to it because you cut it off. So you could actually switch this and turn this off. So if it was turned off, that would basically mean that you had removed the apical bud. Then what we see the auxiliary bud force them more. And if that bud's working more, what type of growth are you going to experience? Lateral growth. So why would you trim the hedges at your house? Like, why would you take the little saw thing that's electric and you go off the top, right? You never really see them go in the middle and do like chops in the middle, just straight off the top, and they're done. Because they're removing what meristem? So once that apical meristem's gone, the only one that's left is lateral, so the growth is outward. You've seen that before. Right now you just finish on the process on it. You've never done that before? Good for you. You're removing a meanophyte. You're cutting off its sexual organ. We cut it off because it was pretty. It's small, you think. Well, it depends. Like, a lot of it will, in some degree, and you're supposed to cut off at some length. And that has to do with the fact that, like, there's, like, a length where you cut off. And you're supposed to cut it, I forget. It's either, like, on a rose bush, you're supposed to look for, once you cut off the flower, you look for the next, like, node that it has a connection point, and that connection stem of, like, 5 leaves. And I think you cut right below those 5 leaves is the way it is. And that's because it's a first stem, like, say that flower is, like, right here. And there's all these little stems that are shooting off of it. You look for the one that has 5 leaves, and then when you cut right below it. So let's say there was 5 leaves, like, right here, cut right below it, stop it. What's left, though, on the bottom is going to be an area where you have, like, a certain amount of meristematic tissue. And it's something about that designation of 5 leaves there that just is, on average, has a level of meristematic tissue that's still fairly apical. Does that make sense? So a new flower bud is likely to form in that region. Now that's not tomorrow it will form, but it's more likely to form than if you cut it off at different spots. That's why cutting the flower off like right at the receptacle, which is where it's connected to on the node, it's like you won't have a flower grow back there that year for sure. Waiting and cutting it back a little bit, like your chemicals are in high concentration as you move away from the flower. It's kind of cool stuff if you think about it. Let's see. Let's talk about growth in woody plants. We still know that there's an apical meristem, right? And that apical meristem is, of course, always utilized for what purpose? Growth of the tip upward, right? Upward growth. All about this quest for sunlight. Now, if you have like a lateral meristem, you know that's growth outward, correct? Now, when we think of outward, like I think people are thinking about like a stem just like popping out of the actual trunk of the tree, and that's not what we're talking about. We're talking about like the tree trunk gets larger in its like diameter in a way, circumference increases. Make sense? So you have actually lateral meristems that are responsible for this, and that's actually adding to areas. You actually have two like types of lateral meristems. You have vascular cambium, and then you also have cork cambium. The cork cambium is responsible for making bark, whereas your vascular cambium is all about making vascular tissue. It's going to make the secondary phloem as well as the secondary fountain. It's the secondary xylem that actually is what becomes the wood, essentially. You think about it, it's a vascular cambium that actually represents the tree ring, or what we consider stems. Okay? Fun stuff?
So when you cut down a tree,
the tree, well, see, it's unlikely to, because the deal is that whenever like a little baby seed is planted and you start like doing this process of like creating a new creature, like typically there's always a little prepackaged leaf that is included with this. Because it's a trunk, there's no leaf at all, and there's no leaf that's prepackaged. The leaves are all gone. So it's unlikely it will survive. Now, it could survive if you had like at least some leaves left. You know, like if you leave a little bit, there could be some survivability. But just a trunk that's been cut down a foot off the ground without a leaf. Now, it's going to still try to do some of the plant actions, but with no photosynthesis coming in, it's not likely to last so long. Let's talk about hormones. And it's hormones in plants. There's auxin. There's cytokinin. There's gibberellin. There's abscisic acid. And there's also ethylene. I feel like I'm missing one now. Auxin is basically for general plant growth. For me, whenever it says the plant's growing, this is the go-to answer, auxin. It could be growth in a variety of different spots. Cytokinins are going to be about making new cells, so it's going to be about cell reproduction. What about gibberellins? What do they do? What do they stimulate? They stimulate seed germination. Anyone know that over here? What does abscisic acid do? I'm glad we're going over these. Seed dormancy. I hope you all study these tonight, right? Travis, you might want to tell your little buddies in fourth grade to study these too. Okay. And what does ethylene do? It ripens fruit. Definitely study these guys. Definitely. Super important. Is ethylene. Let's see. No, we're not done yet. Let's talk about nitrogen uptake. That occurs where? In the where? In the leaves, right? Perfect. Where? Nitrogen uptake. Leaves, right? Perfect. It's gonna happen right now, dude. There's your root right there, right? What type of root have I drawn? Is this a fibrous or a taproot? Do you see? Taproot. Does that mean that this is a dicot or a monocot? It's a dicot. What type of nitrogen would you find in the atmosphere? Ammonium, nitrate, ammonia, and 2. Diatomic nitrogen. Do you know about what percent this represents of your air? About 70%. So a ton of it. Well, how many bonds would you have between 2 nitrogens in a nitrogen diatomic molecule? Triple bonds. 3. I'll just stretch this just like that. How many electrons do you see shared off between these 3 bonds? 6 electrons. 6 electrons in the 3 bonds. Each bond is 2. Which would be easier to break? The bond here or the bond here? The fluorine bond or the nitrogen bond? Because there's just one thing to break. It's simple to break. Nitrogen can do it with this book. We use about 10% of the world's energy breaking this bond each year. That's a fact. Now, we could do this, or we could let a bacteria do it. So, you have these bacteria, some nitrogen-fixing bacteria. And what they're going to work to do is, do what? Take your nitrogen, this diatomic, and they're going to convert it into a usable form. What's that usable form? So it's NH3, which is ammonia. Is ammonia an acid or a base? It's a base. Good. Is it a Lewis base, or is it a, actually, no, it's that question. It's all bases. It's going to be all definitions in one. Now, what could happen to this ammonia? Because typically, how are you taking things in to your roots that are along the way here? You remember the mechanism that takes in stuff? It's like cation exchange, right? So it's basically charged items that can be taken up. Charged ions could go in. NH3 is not charged. So, between the nitrogen fixing bacteria and then the decomposition bacteria, the decomp bacteria that are taking stuff instead and converting that into the ammonia, the addition of hydrogen ions, like from the results of respiration, which of course you would see like in plants, the addition of those hydrogen ions right through there, forms ammonium, which is NH4+, and perhaps ammonium could be taken in by the plant root because it's charged. That's why ammonium couldn't be taken in, it was not charged. Cool? There might also be some other bacteria called nitrifying bacteria that are able to take your NH4 and convert it to NO3 which is nitrate, and then incorporate the nitrate. Cool? In the event that you have like way too much of this NH3 and then maybe probably the NH4 plus and the NO3 then you have some denitrifying bacteria which are essentially going to be able to take all of your unique forms of nitrogen and then reconvert it back into N2 essentially. What type of relationship is that? Symbiotic? Predation? Competition? Symbiotic? What does symbiotic mean? Close? Close by? These bacteria a lot of times live on the roots. Would that be out of the symbiotic ones? There's like mutualistic, commensalistic, parasitic. This is what type of reaction? And that's because not only are the bacteria like giving something to the plant, but specifically the plant's going to be feeding the bacteria. So it works out moderately well. Good? We're not texting, are we? Perfect. What other questions might you have? There's a little bit of like talk about mycorrhizae. Do you remember what mycorrhizae is? No bacteria though. It's a fungus. What does the mycorrhizae do? Like, what does its like focus? Aside from like being on. Mycorrhizae. What? What? What is mycorrhizae? You said it was a fungus? What does it do? Right. Increase the surface area. What's wrong, Julio? What does that mean if you have more surface area? Yeah. Probably what two things? If you're talking about roots, which is where you find this at. Definitely water, what else? Minerals. Yeah, nutrients. K's, magnesium, all those things. Cool? So that's really good stuff. Well, the last thing to mention, I guess, that I hadn't mentioned in the other video from the first period is the concept of double fertilization. Neat. What does double fertilization mean? There's two eggs, right? Let's remind ourselves with an angiosperm, what's the male part? The stamen or the carpel? The stamen's the female part, right? Yeah. The stamen's actually the male part. It's made of what? A couple things. The anther and the filament. Definitely want to study these, right? This is good stuff. The carpel's the female part. What's it made of? I don't even like botany, guys. The sigma. What else? The style. The ovary. Perfect. Which one of these is developing into the fruit? The ovary, right? Like the actual encasing of the ovary is what encases the fruit it's developed into. Basically. Fertilized polar nuclei of the ovary. So that brings us to the next point, which is maybe like a quick picture of what's going on here. So I'm drawing a picture of the ovary. At the bottom here, there's your ovary. So this is the sigma, right? or the style. The stigma's at the top. And then you drop off what at the very, very top? A pollen grain, right? Now inside that pollen grain is a lot of things, but there's actually there's three major things, I think. One is going to be the tool that could actually start to form this structure. What am I drawing here? Pollen tube, perfect. So I'm drawing the pollen tube. The pollen tube is formed, doesn't naturally exist, it's formed. The tube is formed by the pollen grain. And then there's a deposit of two sperm. There's sperm 1 and sperm 2. Those are little stars that travel downward. The first sperm that enters in is going to fertilize the what? The egg, which is what makes the what? The seed, the zygote, basically. And then the second sperm, it's going to fertilize what? The polar nuclei, which themselves are what? 2N. So when that happens, what do you form? 3N endosperm, and it's that you eat. So the endosperm is the 3N tissue that you ingest. That's what you eat. So when you eat a fruit, and you think meat, you're eating triploid tissue. It's kind of different to think about. But then again, you're all good. Good? Questions about this? Because a lot of fruit, that's a good question. When we think of like a flower, a lot of times we think of this structure like existing one time in the flower. Like, you see like the rose and you see like one of these. And it's very typical that you have one of these guys and then you have like many of the antherfilament combos, which is like the male part. But there's a lot of plants out there, flowering plants, where there's multiple of these guys, multiple of the carpels that you see here in one single flower. Does that make sense? So in one flower, this female part is reproduced many, many times. Well, each time one of these little ovaries is fertilized, it becomes a fruit, essentially. Well, when they're very, very close together and grouped up like they are in one flower, these ovaries are all going to fuse together and form something that you would know like as a raspberry or a blackberry. You ever seen like those fruits that they have like the little bumps in them almost? That's because you've done like many of these ovaries together. Or a lot of times, sometimes, there's actually like many of these and many anthers, many of these and many stamens. So your differences in multiple seeds versus not multiple seeds is that fruit made of like one ovary, or is it made of many of these ovaries in a very, very close proximity? Right, essentially. And see, the benefit of that is that now you put a guarantee that you don't just like produce one apple with one seed, you produce one apple with many seeds, more opportunity. Question? I really don't know much about oranges, but they're from a tree. I know that much. I don't really eat them. As far as like their orientation, they're going to be a unique type of fruit. Let's see. I think there's multiple seeds in them, right? Typically is. That would mean that they have multiple carpels. Like whenever this fusion happens, there's going to be multiple carpels there. It has to be. On their outside. A lot of times the ovary, will differentiate. The tissue on the outside becomes different. Like when you look at like an apple tree, like the ovary that exists before fertilization doesn't look the same as like the outer part, if you will, of an apple when you buy it in the store. It doesn't look the same. So there's differentiation on the inside and the outside as well.